Hello, and welcome to the BYU Family History Library webinar series. We're glad you could join us today. I'm Bryant, and I'll be your host for this webinar. Please make sure your microphones and web cameras are disabled during the presentation to provide a smooth recording. If you have technical difficulties during the webinar, please use the chat box and I can address your concerns. You're welcome to use the chat box during the webinar for comments and insights. However, all questions will be addressed at the end of the presentation. Our upcoming webinars are on the 13th, when one record isn't enough, building a complete picture with multiple records with Catherine Grant, and that'll be on Friday at 4 p.m. And on the 18th, we have 2 steps forward, 1 step back. Is this genealogy or the cha-cha? With Sarah Cochran, Wednesday at 4 p.m. If you'd like to access previous webinars, please visit our webinar index on our website or search our YouTube channel. All of our webinars are recorded and uploaded by the following Monday for your convenience. We also post links to recordings and other updates on our Facebook and Twitter accounts. For today's webinar, we are pleased to hear from Bonnie Belza, who will be presenting on DNA Kindergarten Part 1. And the rest of Bonnie's parts will be on the upcoming Saturdays at 4 p.m. as well. And you can find a schedule of that on our website. Bonnie Belza is a family historian and genetic genealogy DNA researcher whose degree in gerontology led her to genealogy research. She has attended I4GG, the International Genetic Genealogy Conference, Roots Tech, SLIG, and many national local conferences, and is a frequent lecturer. She teaches the DNA kindergarten class at the 500-plus member West Valley Genealogical Society in Arizona and presents on DNA genealogy with a focus on learning how to use DNA fun. If Bonnie is ready, we'll turn the time over to her.
Well, thank you for that introduction, Bryant. This class is called DNA Kindergarten, and we're going to have a 4-part webinar. It's been designed for those of you who may not have learned about DNA in high school because it wasn't taught until the late 1960s. It's also for those who might not be familiar with building a family tree for genealogy. Both of these groups of people may have taken a DNA test and want to know more about it, and you may be able to help them if you're familiar with both of those. If we can have a quick poll, how many of you have taken a DNA test? Just in the chat box, tell us. I see there's some people that have and some people that haven't. All right. Well, we are hoping that even if you've taken a DNA test already and have dived into it, that this will give you a way to understand it a little more simply or be able to share your understandings with other people. Learning something new can be overwhelming. So we're going to take a small focus and stick with the basics. Sometimes when you're learning something new, you can feel like a failure. And so we're going to flip failure on its head and learn from it. Sometimes you can feel like you're treading water. So I want you to find enjoyment in the process of learning about DNA, like you did when you first learned to swim. It can also give you a feeling of uncertainty. So relish the uncertainty and let's get started. We're going to start with something that you know, your family. Whether you've done genealogy before or not, genealogy can be used as a way to document and track information on your family. Now, one of the things that always bothered me when I heard someone speak about genealogical topics is they would use their own family. We're going to be taking a look at a family that I call the Smith family, and this is our Dick and Jane family. So mom and dad, Dick, Jane, and Sally. They can demonstrate for us different genealogical relationships. So we have parents and spouses, and we have their children, who are siblings. This is a 2-generation descendancy chart, and you get your DNA from these direct biological relationships. Here we're showing a pedigree chart. In this one, you're on the left side of the screen with your sibling and spouse, And then we work back in time with your parents, your father and mother, and your paternal and maternal grandparents. I've abbreviated because it doesn't quite fit in the box. It's my paternal great-grandfather and my paternal great-grandmother. You have 2 parents, 4 grandparents, and 8 great-grandparents. How many great-great-grandparents do you have? That's right, you have 16. It doubles with every generation. Now here's another family that you may be familiar with, the Cinders. Cinderella's father remarried after her mother passed away, so she has a stepmother, and her stepmother brought along and had 2 children with her father, Drusilla and Anastasia. The Cinder family allows us to look at genetics versus genealogical family relationships. Cinderella, Drusilla, and Anastasia are half-siblings. Cinderella doesn't share any DNA with her stepmother. They're not genetically related, but they are a family. You only share your DNA with those from the same biological lines. And take a look at this family, Snow White and her group of dwarfs. How are they related? We don't really know. Doc and Grumpy may be brothers, or they may be father and son. DNA will help us figure out relationships when we don't know how people in a family might be related. During this webinar series, we're going to look at 4 different topics. We're starting out tonight with the science of DNA. We'll continue with an understanding of the ethnicity that we can gather from our DNA. And then we'll use the puzzle pieces of DNA and the relationships to verify and expand your family tree, and use shared matches, finally, to break down brick walls. Let's get started with DNA science. Whenever I want to learn something new, something I've found helpful is going to the children's section in the library and grabbing a book about a subject. It's a basic understanding of the subject that I'm interested in. The lessons here on DNA science were taken from the Duxter's website, which teaches science to children. So what does DNA stand for? DNA refers to deoxyribonucleic acid. DNA is an essential molecule for life, and it acts like a recipe that holds the instructions telling our bodies how to develop and function. You've probably seen this picture of the double helix. DNA is a long, thin molecule. It's made up of something called nucleotides. There are 4 different types of nucleotides: adenine, thymine, guanine, and cytosine. They're usually represented by their first letter, A, T, G, and C. Holding the nucleotides together is a backbone made of phosphate and deoxyribose. So nucleotides are sometimes referred to as bases, and when they form the rungs of the twisted ladder, they are referred to as base pairs. The DNA code is made up of these 4 base pairs. They're grouped by 3s into codons, and a string of codons may look like this: ATC, TGA, and be repeated. The different letters represent instructions. Our cells read the instructions on the DNA. Even though there's only 4 different letters, DNA molecules are thousands of these codon strings long, and it allows for billions and billions of different combinations. This is the code that's used to represent your raw DNA. Chromosomes are long chains made of DNA and protein. We have 23 pairs of chromosomes and 46 chromosomes in each cell, 2 times 23. One pair of chromosomes, the 23rd chromosome, thus the company 23andMe, is referred to as the sex chromosome. It determines your gender. If you have an XX chromosome on 23, you're a female, and an XY pair is a male. All the other chromosomes are numbered 1 through 22. This is a picture of our DNA chromosomes called a karyotype, and it shows 23 pairs from the largest to the smallest. Can you tell who this DNA is from? That's right, it's from a male. Notice that the 23rd chromosome is an X and a Y. There are three kinds of DNA. Mitochondrial DNA is found in the cell mitochondria, and it contains genetic material from the mother. Y DNA is found in the nucleus of the cell. Only males have Y DNA, and it's passed from father to son. Autosomal or AtDNA is also found in the nucleus, and we get our autosomal DNA from both parents. Mitochondrial DNA is passed from mothers to their children, but only women can pass it on to their children. So it's a documentation of our maternal line and traces back for 150 to 200,000 years. Most often, it results in all the woman's descendants in a maternal line sharing the exact same mitochondrial DNA. Mismatches in mitochondrial DNA can rule out a relationship on that line. Mitochondrial DNA can also be used to identify a biographical origin for your maternal line. Y DNA is also found in the cell, in the nucleus, but only males inherit Y DNA. And as I said earlier, it's only passed from father to son. So it's a record of our paternal line, which sometimes aligns with our surnames. It traces back 60,000 years. And like mitochondrial DNA, mismatches can rule out a relationship in the Y DNA line. Y DNA can also be used to identify a biogeographical origin. Y DNA and mtDNA use haplogroups to identify themselves. So a haplogroup will tell you which type of Y DNA or mtDNA you have. It makes reference to a specific set of shared genetic variations. In this series, we're focusing on autosomal DNA. AtDNA is what we work with most in DNA analysis for genealogy. We inherit it from our parents, who inherited it from their parents, in an entirely random fashion. Recombination occurs when our chromosome pairs are separated and come back together to pass on to our children. The resulting blocks or pieces of autosomal DNA are called segments. So who did we get our DNA from? Well, as we said already, your mitochondrial DNA comes from your mom, your Y DNA from your dad, and your autosomal DNA from both parents. But since your parents got it from their parents, you have DNA from your ancestors. And autosomal DNA can look back into your ancestry 5, 6, sometimes 7 generations. So our DNA matches, who have DNA shared with us, provide clues to these distant relatives. Centimorgans are a measure of genetic linkage, and it's one of the ways we describe our DNA pieces that we share with someone. The number of centimorgans that we share tells us the likelihood that a match also shares a single recent common ancestor with us. All of the DNA testing companies report the amount of shared DNA as a number or as a percentage of centimorgans shared. All of the companies report the total number of shared centimorgans. So how do we use these centimorgans or pieces of DNA? Well, we want to know how much is shared in centimorgans with each of our matches. Since we have about 7,000 pieces to start, we want to know how many of those pieces we share with someone else. We want to know how big the shares of pieces are. That measure is described in terms of how many segments we have with that person. We want to know where we got the DNA from. The where is used to determine what population we came from. And who we got it from is determined from inheritance. So while we inherit 50% of our DNA from each parent, due to this recombination process, siblings don't inherit the same 50% from each parent. As you see on the father's side, he has two blue segments. Those break apart and they're recombined to create the segment passed on to their children. And so while both siblings here receive 50% of their DNA from their father and from their mother, they didn't get the same 50% from each parent. Siblings will not share the exact same DNA. Also, because the testing companies use different testing chips, we can be shown to share different amounts of DNA, slightly different, when we test the same person at each one of the testing companies. This is my maternal aunt, and at Ancestry, she shares 1,897 centimorgans with me. But at Family Tree DNA, she shares slightly less, and at MyHeritage, slightly more. I want to thank MyHeritage here for their new colorizing capability so that I could colorize Aunt B from the Andy Griffith show. Before we move on with analyzing these pieces of DNA described as centimorgans, we need to figure out who we might share them with. And to do that, we need to understand the relationships between relatives beyond the traditional grandparent, great-grandparent, and straight-on back relationships that we are already familiar with. So take a moment now and take a piece of paper out and write a picture. Who is your first cousin once removed? Got it? Well, in case that's not a concept you're familiar with, here's a picture of it. Your first cousin once removed are the children of your first cousin. There's a generation between you and that person, and so they are once removed. Another way to understand the cousin rule is to use the G rule. The number of G's when you describe your grandparent relationship between you and a cousin equals the number of that cousin. So our first cousins share our grandparents as a common ancestor, 1G, first cousins. Our second cousins share great-grandparents as a common ancestor, 2Gs, second cousin, and so on. And again, the number of removed that a cousin is from you indicates the number of generations separating you. Well, now that we have an understanding of relationships, how do we figure out what we share with each one of our relatives? On this diagram, you can see the statistically predicted relationships. So, for example, you share 50% with your father and 50% with your mother. You also share 50% with your siblings and 50% with your son or daughter. You share 25% with your grandfather and your grandmother, and 25% with your niece, nephew, and your aunts and uncles. You also share 25% with your granddaughter and grandsons. Statistically, we share DNA, shown in this diagram, as little as 6.25% with our great, grand, or great aunts and uncles. But how many centimorgans do you actually share with your relatives? Blaine Bettinger, who is a well-known lecturer and writer in DNA genetic research, initiated a crowdsourcing project, and with the help of other folks, had analysis done of the results of citizen scientists who contributed the actual percentage of shared centimorgans they discovered that they shared with their known and well-documented relatives. These crowdsourced results were used to calculate a range of centimorgans that you may share with a particular relative and the average shared centimorgans for each one of those relationships. That chart is called the Shared Centimorgan Project. This is a handout that you should have had from the video, and it will be available on the website of the video. The way to read this chart is that each box has in the top of it the relationship to you. You're the self in the middle of the chart. Each box has that relationship, and then it describes the average amount of centimorgans that you share at that relationship. So for aunt and uncle, the average is 1750. It also shows the range of centimorgans that may be shared at that relationship. Now, this is really helpful, but when the amount of DNA that you share is a number like, let's say 175, you start looking around the chart, and there's a lot of places where 175 can fall. Wow, how do I find out and identify all those different relationships? Well, a gentleman named Johnny Pearl automated this and provides a website that's available online. This website is at dnapainter.com, and one of the tools is the Shared Centimorgan Project tool. You just enter the number here on the tool. You can also enter a percentage by clicking on this Show Percentage box. And when you enter that number, you'll be given the relationship probability for that many shared centimorgans. So in this example, I entered 750 centimorgans that are shared with a match. And the tool is showing me that I have almost an 85% probability of this person being a great-grandparent, great-aunt, uncle, first cousin, and so on. I'm going to use my information I have about that match to eliminate some of these relationships and focus in on how myself and my match might be related. The bottom half of the Shared Centimorgan screen will show you and by those relationships by highlighting them in the chart. So you can see that at 750 centimorgans, I share all these folded relationships potentially. Now, here's another exercise for you. Pull out your handout that says ATDNA Inheritance. And in each box, I want you to enter the relationship that you have to that person, with you being the person in the middle on the bottom row. And I want you to, from your statistical chart, to fill in the statistical amount of DNA that you would share as a percentage. And then from the Shared Centimorgan chart, list the amount of shared centimorgans with that particular relationship. So going up this tree, this is a sibling, and this is a sibling. This is your father and mother. This is your paternal grandparents. your maternal grandparent, and a great aunt or uncle on your maternal side. This was your mother, so this is one of her siblings and her sibling's spouse. And this person in the bottom right-hand corner is your first cousin. So let's take a moment and, using the charts, fill in your ATV and inheritance records. I know that's not enough time maybe to fill in all the chart. And here's the answer key if you'd like to take a moment to fill in your chart. One of the things I want to point out to you here is a lot of times the testing companies will tell you that someone is a cousin, but you want to use your shared centimorgan interactive website. Use the number of shared centimorgans that you have with a match and see what it comes up with. Because while they may call it a cousin, it could be another relationship. Notice that first cousins share an average of 874 centimorgans, or 12.5% statistically. Also notice that because your aunt or uncle's spouse is not related to you, you share 0 centimorgans with that person. So in today's lesson, we've learned about family relationships. We've reviewed genealogical descendant and pedigree trees. We've answered the question of what does DNA stand for and what is it? We've described the 23 pairs of chromosomes and noted that the 23rd chromosome is the one that determines our gender based on whether we have an XX or an X and a Y. We've reviewed the different kinds of DNA, mitochondrial DNA, Y-DNA, and autosomal DNA. And we've described how to use the number of shared centimorgans to measure and determine our relationships with our matches. We've also described how DNA is inherited and how you can predict the amount of DNA that you should share with a relative. Here's a quick quiz. True or false? My father's brother's grandson is my first cousin once removed. That's true. True or false? Full siblings have different DNA. That's also true. And true or false? Everyone in this webinar has taken a DNA test. Well, you can check the chat bar and see if that's true. Our next installments of the remaining lessons in this webinar series will start at 4 p.m. Mountain Daylight Time. We go on daylight savings this weekend. So we'll have a program on ethnicity on March 14th. The webinar on March 21st will cover how to use your DNA. And April 4th, we'll talk about how to interpret shared matches. If you have any questions, you're welcome to contact me at bonniebelza@gmail.com. And with that, Bryant, I'll turn it back to you.
Thanks, Bonnie. We'll now turn it over for any questions that the audience might have. So if you have any questions for Bonnie, then please post them in the chat box and she can answer them for us. It looks like our first question says, where do you see DNA research helping family history in the next 5 to 10 years?
It is so difficult. That's a great question to predict where this will go. So many changes have come about just in the last 5 years. And I think we're going to be in for a lot of exciting and interesting applications of DNA. Today, it allows us to confirm our relationship in our trees, find people that go back 5 or 6 generations, I can't imagine what other things we have in store for us. One of the things that's a real big focus for some people are the using it for health purposes and to identify genetically related health issues.
Awesome. Great answer, Bonnie. Thank you.
I see when some of you haven't taken a DNA test, the prices are very reasonable on them, and often you can see around any holiday people will reduce the price. When I do the full DNA class, I suggest that people test first with Ancestry DNA, and that's not because of any bias toward them. It's because they're the biggest pond that you can fish in to find matches. They have over 16 million DNA tests in their database. The second largest database is 23andMe, who have over 10 million. And since you can't trade tests between those two companies, I suggest that if you're looking for an unknown parent, or if you are wanting to see all the DNA matches that you might have in the two different databases, that you test in both places. So you can take the results of either your 23andMe test or your Ancestry test and transfer it for a much lower cost to MyHeritage or Family Tree DNA. You can also transfer it to GEDmatch, which while you can't test there, GEDmatch allows you to compare your test results with people who've tested at other DNA testing sites. So it's pay for two, and you can actually get 5 tests rather than buying them at all 5 places.
Great. And it looks like we have one more question. It says, so from what I understand, the DNA test can only currently show relatives up to the third cousin. Is this true?
No, actually, Lorley, it will show you, or can I potentially identify relatives as far back as 5th and 6th cousins. Autosomal DNA can be used back that far. It is more accurate at the closer third, second, first cousin, and of course, direct relatives. But it can identify relatives that might go back that far. You have to use regular genealogical research techniques to find the evidence to support that relationship. But the DNA can identify people that it might be the only way to get that far back when you have a brick wall. Does anyone else have any questions?
Looks like we have one more question. It's from Lee. It says, are there privacy concerns with making DNA results publicly available?
Lee, thank you for that question. All the companies, when you submit your DNA test to them, ask you to agree to their terms and conditions, and they all commit to keeping your DNA information private and secure. So unless you share it with someone on your own, they're not going to be sharing your specific DNA information. They will use it, and you can read about it in their terms and conditions for research purposes in an anonymous, unidentified way. There's also been concern lately about the use of it by law enforcement, and I would recommend watching the, Brian, I'm sorry, it's somebody else's video, but from Roots Tech, Blaine Bettinger did an excellent summary of the use of DNA by law enforcement, and you can find that free on the Roots Tech site. I see there's another question here from Dolly that asked, my grandmother's parentage is in question. Not a lot of people on that site have been tested, but is there a possibility of figuring that out? Yes, Dolly, when you get your shared matches, you will recognize potentially some of those people as being on your father's side and your mother's side. So you can eliminate some people that share DNA with you because they'll be on the wrong side of the grandparent that you're looking for. You can take the next step, and if you can find second cousins that would match you at that grandmother level, you can eliminate those that you know And you'll be left with people that you don't know that might point you to that unknown grandmother. We'll get into that a little bit more in the further sessions.
Great. Thanks, Bonnie. And it looks like there's no more questions. So that will be the end of our webinar today. Be sure to join us next time. I just put up the schedule for Bonnie's next webinars. The next one's on March 14th, and that'll be on Saturday, not on Friday. And that will be part 2 at 4 p.m. And as well as the upcoming Saturdays after that, we'll have Bonnie's webinars. And also be sure to join us next time on the 13th, and that's on Friday. And that's with Catherine Grant, When One Record Isn't Enough, Building a Complete Picture with Multiple Records. So we hope to see you next time. Thank you, Bonnie, and thank you everyone for joining us, and we hope you have a great day.